at that point, I was kind of a broken postdoc. Once you're behind, it's really hard to catch up. And if you were to put your CV out, people would be like, there's no way we're going to take this person. They haven't published and kind of broken. And Charles asked me what was my plan. And, I was like, I'm going to get a job. That's my plan. And I didn't have any prospects, right? I had applied at that point for almost probably 100 industry jobs.
Okay.
I had two real interviews there. Kind of had just said, okay, I'm going to give up this academic dream and do industry. And then came here and things, 180'd.
Hello and welcome to another episode of The Next Step. I'm Jeff Kozlowski, your host. Today I'm excited to welcome John Lueck, who is Associate Professor of Pharmacology and Physiology here at University of Rochester Medicine. Got his PhD here in 2008. We're going to talk about his graduate school journeys, all the potholes that maybe we run into afterwards, and the full journey of having a career in academia and science. John currently focuses on finding new therapies for genetic diseases like cystic fibrosis and myotonic dystrophy. My co-host today is Charles Thornton, Professor of Neurology. Charles, thank you for being another co-host here on The Next Step. Can you tell us a little bit about what you do here at the university?
I came here as a clinician, and I was interested in neurogenetic diseases. And I have a career training path that I wouldn't recommend to anyone else, which is that after getting interested in a particular disease and wanting to work on treatment, I had to diverge considerably to start working in the lab to understand more about the disease, to shape a treatment.
All right. Thank you again for joining. John, let's start by just talking a little about, I forget, are you originally from this area, or how did you discover
It's interesting.
Rochester?
Yeah, so I was actually recommended to come here by my advisor when I was at Bradley University.
Okay.
And at that time, I'm from north of Chicago, between Milwaukee and Chicago. Went to a small liberal arts college or university, Bradley University in Peoria, Illinois. Wanted to go pre-med. But then quickly realized I wasn't good at taking tests and would never get into medical school. And so I liked problem solving. I liked teaching. And so doing research at an institution seemed like the next best thing. And so my advisor, Eric Stabenow, he recommended going to University of Rochester because of their strong background in kind of physiology, and also the idea, he saw that I was really interested in kind of translational research. So yeah, I interviewed at a bunch of schools, and this is how I ended up. Little did I know, actually, because I didn't really pay attention to it, my dad got his PhD here. So when I told my parents I was coming here, my dad's like, I got my PhD. I was like, yeah, I guess that does make sense. This is where he grew up. So I used to come here a lot as a kid. So I didn't pay attention much to the details as a kid, as a kid, teenager, maybe not even now. Yeah.
And so, do you remember anything about kind of first impressions when you got here, whether it was interviewing or when you first came to Rochester?
Yeah, I mean, it was the first place I interviewed where I felt like it was home. Huh. I interviewed maybe at 9 other programs, and I really felt like the researchers here spent a lot of time with trainees, and that was really important to me. And also, it turns out that I interviewed with the person who is now my wife, so that helped a great deal. I met her on the bus, and so we've been together ever since. So
I'm gonna have to get you on like a special Valentine's Day episode of this.
That sounds good.
At some point. It's so interesting. Bob Dirkson's name comes up a lot, and maybe that's just because we've interviewed several Pharm Phys folks along the way, but Bob's name comes up quite a bit in conversation. Always great things. Can you talk about just kind of your early interactions with Bob and how you kind of got hooked up with him?
Yeah, so I came in through a program that doesn't exist anymore, and it kind of transitioned to the Cell Molecular Pharmacology and Physiology program that now exists as a PhD program. And Bob was pretty new faculty member at the time. His lab was pretty small, but he was always out in the hallways talking with trainees, and he was really into mentoring. When I was looking to do research, I actually thought I was going to do the save the world science. I was interested in cardiovascular research, and but then quickly realized that maybe wasn't the most important thing. It was to hook myself up with somebody who could teach me how to do research. And it was really when I sat through one of the classes that Bob Dirksen taught, and I really liked his style. He was funny, kind of self-deprecating, but was really into the details and mechanistic on how things worked. And so I talked to him after the class and asked if I could rotate. And it was something I had no idea about. Ion channels, I didn't really know much about those. I didn't really think too much about skeletal muscle physiology, but I liked our interactions. So that's how I picked that lab, and that's how I ended up going there. And it's been great.
And how did you end up focusing on myotonic dystrophy eventually?
So that was completely luck. So Bob had another graduate student in the lab. We started at the exact same time, and Bob's like, I have some projects. Which ones do you want to do? And the other student kind of called dibs on what was the bread and butter of the lab, which was excitation contraction coupling, looking at diseases that are caused by mutations in the RNA receptor, and so he was like, that's mine, you get the other one. And the other one was myotonic dystrophy.
Okay.
But I really think I lucked out. I think that's was the best happen chance kind of situation that could have come across.
And Charles, not too long after that, that's where you come into the picture because of Bob Dirksen, right? Bob kind of saw something and thought you two might make a nice match. What was that story all about?
Yeah, it was definitely through Bob that I first met John. And I don't recall the particulars of how we started working together, but pretty soon there were occasions where we were both hands-on working in the lab on the same thing over extended periods of time and having long conversations and getting to know each other, enjoy each other's company, but also the scientific interchange. And I think that's what launched it.
Yeah, and what I realized I was really lucky is this idea that took a path that Charles was mentioning, took a path that he wouldn't recommend. But it was really this boots on the ground, understanding the research and doing the research, that he knew how hard things were, and so expectations were set that were realistic, and ideas of what can be done, what couldn't be done, were ambitious but reasonable. And I think coming in, I didn't really know what to expect. There was a clinician, wearing a white coat, but it was really when we'd have our discussion, it was like sitting next to a postdoc, because he knew all of how to clone, and he knew about bacteria, he knew all the things. And so it was nice, and I kind of knew that was going to be a really good relationship.
How did those conversations with Charles spark an interest in molecular genetics and disease mechanisms?
I think it was kind of the way that was presented to me. It was, you had these. So Charles, as a clinician, talked about the patients first, and then stepped backwards, and then got all the way back to what was then kind of recently discovered of the mechanism, the root cause of this disease. And I liked that. I liked the idea of interrogating the mechanism of disease. Once you figure that out, you say, okay, well, these are the targets that we can hit with a therapeutic. And that, to me, seems like how Charles has worked through his career, and it was successful. I feel it was kind of amazing. The experiments that we did really early on were kind of amazing, and they worked really well, and it gives you motivation to continue to do more, right? So, yeah.
Anything else you remember about those early interactions, early conversations?
This does make me recall what was happening in those days. And I had actually come here to train in experimental therapeutics, developing treatments for diseases that didn't have treatment yet. And I got interested in a particular one. And in the environment that I was working in, the clinical environment, I was kind of surrounded by people the conversations went like this. Well, I wonder what would happen if we gave them X or Y or Z. They were kind of loose thinking therapeutically, and these were all long shots that were destined to fail, and many of them were tried and did fail. And so I had this perspective that that wasn't going to be the path that I wanted to follow. I wanted to try and understand things at the root cause and then come at them more systematically. And it was the interaction with Bob Dirksen and John that originally allowed us to kind of pry the door open well enough to kind of see root cause and then think of strategy to get at the root cause. And at that point, it's surprising how quickly the process moved from just first experiments to kind of test out something that was a bit wild and crazy to things that actually were starting to work pretty effectively.
Yeah, and I was really eager. So I wouldn't say very smart, but I work very hard. And so, trying to get these experiments to go. Back in the day, I would often work 36 hours trying to get these things figured out. And it paid off. It was fun to do those things. I wouldn't recommend that to most people, but it was and it kind of, there was a lot of meaning to the work that was being done as well. So
yeah, we've had a previous guest on. Her name is Bohoon Lee. She's a child neurologist here at U Rochester, and during her conversation she talked about we talked a lot about, she does a lot of clinical trial work and was in, not something you get a lot of training in medical school, but was interested in it. And we talked like, how do you insert yourself into that? And, she mentioned, and now she works very closely with Emma Scheffaloni, another child neurologist here. And she thinks that Dr. Scheffaloni appreciated her, just like you mentioned, that eagerness, the
tenacity.
Like this person's going to show up, they're interested in the work, and that, I think then helps kind of the clinicians and others that may not have had a chance to do these things before or hadn't thought about it, in this mentorship way before, but like kind of helps keep you excited maybe.
But also it was two ways, right? So I'd get results, I'd get excited about them, I'd bring them up. Charles was a super busy person.
Yeah.
But I always found time to sit down and look at the results and say, well, what if we did this? What if we did that? Can you explain this to me? And like really, treated me like a colleague. Mm-hmm. And I really think that that's important. And I think that's really important for training.
I was going to ask that next. So you mentioned that in previous conversations that it felt more like a colleague than a student with Charles. How does that impact your confidence as a trainee or,
Oh, it's huge.
Yeah.
Yeah. You've been to many meetings. There's always famous people at these meetings that you maybe as a graduate student or postdoc or even as a faculty member you want to go talk to. And there's people who are always looking for the next famous person to talk to. Like they're always trying to advance their career through interactions. And I think that who I've really appreciated over the years are people who are just happy to have a good conversation no matter who it is. And, and I'd go to these meetings and, interact at poster sessions, and I cherish those. I think it's super important. And I think that, it has been important for me.
And did you do a lot of that as a trainee?
Oh, yeah.
Yeah.
Yeah, I did. I didn't really care if somebody was a Nobel laureate or anything. I wanted to ask them a question. It wasn't just to talk to them. I was just curious about stuff.
Yeah.
And, and I've been denied conversations because I was just a graduate student or I
really?
was just of course. Yeah.
Yeah.
Or they'll talk to you, but you can tell that they're gazing over the horizon for the next person to interact with. I mean, you've experienced this.
Not fully there.
Yeah, exactly. Yeah. And it's those people who are really engaged and, it's really, really rewarding.
Let's go to then, coming up on the end of your time at University of Rochester. What in your mind was the next step?
I had a really quite successful graduate career. Mm-hmm. Published a bunch of, I thought, was pretty good paper. Learned a lot. I honed my skills in problem solving. Kind of knew what it took, and then it was off to do a postdoc. and, kind of leave the nest. And that's the first time you got to say, oh, what I learned, do I really know what I'm talking about? And because, the way I always expect it to be is I'd show up in a lab and they're like, all right, here's your project, get going on it. So I asked people, like, where should I go to do a postdoc? And I got really, I think, terrible advice.
Wow.
I think there was this, you should go to a really famous lab.
That was the advice.
Yeah. The more famous the lab, the better off. And so there was, try to find HHMI investigators, try to find people that are at really good institutions, and hook your wagon to those people, and you're going to end up with the big 3 paper, Nature, Science, Cell, and that's all you need to kind of take off and start your career. Now we can get into the next bit of it, and so I did follow that advice, and I interviewed at some top-notch labs, and they're good labs, producing, doing really good science, but what I didn't take into account is fit, and like how I approach science, what I need to be successful into that. And so, what I realized is that when I was at Rochester, I could walk down the hall and I could talk to anybody, and people were thrilled to talk about research. Charles, Bob Dirks, and other graduate students. And I didn't take that into account. So when I picked the lab that I went to, it was sold as, you're going to get in 3 years, you're going to get your Nature, Science, and Cell paper. You're going to go off and you're going to get a job as an assistant professor. And I was like, okay, that sounds great. And I didn't really look into the fact that, there were 13 other postdocs in that lab who had the same dream that I had and hadn't achieved that. Okay. And I think it's just really important. I mean, and it'll work out for some people, but if it doesn't, where are those people? And that I think is the most devastating part of academia, is that it's very unforgiving, and you're always are shooting for the moon, and sometimes you miss. And I think it's better to keep building your toolbox and you're bettering yourself and not taking pot shots, kind of like your idea with therapeutics, right? Just throwing them out there. I think it's good to just build and understand, and you'll be successful if that's what you do.
Were there any indications, like looking back, was it hard? Do you feel like you were maybe like blinded to the specific idea of what it's supposed to look like? And looking back, like through your interview processes, could you have seen any of that?
No,
no,
I don't think so. No, maybe, I don't know, but it happened. Yeah. And I quickly realized, maybe a year in, that this was going to be a tough road. I went in with really high stock. I could have gone to many labs, and and then I went through some times, but I learned a ton.
Anything you would say? It sounds like a wrap-up question. We're not at the end and this is for both of you. I think trainees and just a general difficult lab environment. What do you say to them?
You should always be kind of taking a step back and be like, is this going the way that I thought it was going to go? And there's this idea of like, and I have this, and is that I'm not going to give up. I'm going to continue going, and I'm not going to change my course. And this is this idea we've discussed about throwing good money after bad money. You're not a failure if you switch gears. I think that's something that people need to realize. I've been successful, especially since coming to Rochester, coming back, and we can talk about that later. But, I think about this time to time. It's like, would I do it over again? And it's like, sometimes I say yes, and sometimes I say no. I gave up a lot to get here, and it was hard. Yeah, and it was a lot of just grinding and working and moving goalposts and to be successful. And at the end, it all ended up being a simple interaction with Charles.
Yeah,
that got me back here, right? So, but that being said, I did 2 postdocs. The first one, I had one maybe middle author paper in 5.5 or 6 years, but I did learn a lot. I learned all these different techniques. I was very self-reliant. I learned how to do big science, so, these not, like big science, like big idea science. And one thing I learned was how to do like the jugular experiment first, the one that's going to totally deep six your project. You try to do that as early as possible. Some people are afraid of the answer. I want that answer really early on. So I did learn a lot of really crucial, like really important things about research, but I think my time could have been better spent doing something else. Yeah.
Do you, before we jump into getting back this way, is there anything, if someone is interviewing for a postdoc, or is there anything to like look out for or just be cognizant of? Or is it really hard to say, is it something you just don't really know until you're in that environment?
I think, no, I mean, I didn't look at the most recent success of the postdocs that were in that lab. I didn't ask about inter collaboration. I think that it's really important to be in an environment where you can talk to people and discuss their science and learn from them. And like, how do lab meetings go? Like, I think that's a really important thing. So do you have lab meetings every week? Who presents? How do those go?
What does a good lab meeting look like?
Well, I think it's different for everybody, but for me, so like the way I run my lab now is based off of my experiences. And so when I was a postdoc, it was in a big lab, and you would present maybe once a year, because it would be every week someone would go. But then, when the PI's out of town, you wouldn't go that week. And so you would go a long time without presenting any of your data or getting any feedback on it. The person was around to talk one-on-one, but I think it's good to get this kind of community. Science research is done best in a community, right? So the way I do lab meetings now is everybody in my lab presents every week. And it keeps people engaged. You're not getting all of your data just before you have to go present. And it keeps you, I think research is a marathon, not a sprint, and it should be done steadily and not in spurts. And so that's really changed the way I run my lab. I think being able to talk science with your colleagues is super important. Do you go to meetings? How does the paper writing process go? And where did your last 3 mentees end up, and when was that? There's a lot of these questions that I was like, kind of, I didn't think to do that. I figured that person's super famous. They did that because they're famous because they did all the right things, and that's not, and it works out for these people, and they're really good scientists, but it doesn't always work out for the postdoc or the trainee. Yeah.
All right, Charles, we'll bring you back in. John eventually came back to town for a talk, I believe.
Yeah.
And you guys had dinner. What did that dinner look like? What did that conversation look like?
Well, actually, we had a conversation in your office.
Oh, it wasn't dinner. I'm
Yeah, it
sorry.
wasn't dinner.
An office convo.
Yeah.
We picked up where we had left off, and there.
That probably felt good.
It did, yeah.
Some years had passed in the interim, but John had kept up with everything that was happening in the field that he had left behind in Rochester. And so it was like he'd never gone away. It was also like he picked up a whole lot of new stuff.
Yeah.
And he was still a very energetic person and a brilliant thinker. Wow. And so it was a lovely conversation. And right from the start, there were ideas that were popping up that made us both feel very excited, actually. I don't think it would have been essential for that meeting to have taken place for us to have heavily recruited John back to Rochester. But it did help that we had it. And there was, at that point in time, there was no particular plan to expand our faculty. So we had to go out and try to figure out a path to bring somebody on board, because it was a rare opportunity and we didn't want him to go anywhere else.
And from your perspective, what did things look like, and what were you thinking?
I was a little bit different from my perspective, maybe, but it was an amazing conversation. It was at that point the best scientific conversation I had in years. It was like, when you see two twins talk to each other, they kind of understand what they're yeah, you have twins. You get they kind of understand each other, and I felt like that. It was really fun. Like we were using the same hand gestures to explain really complicated problems, and it was fun. It was just fun. And at that point, I was kind of a broken postdoc. I did a second postdoc, which was much more successful. So, published a bunch of papers, but at that, once you're behind, it's really hard to catch up. And if you were to put your CV out, people would be like, there's no way we're going to take this person. They haven't published, and kind of broken. And so my thought then was, if I can get myself out to give talks, I can show that I kind of know what I'm talking about, and I'd be a collaborative colleague in this and that. So actually, I did while I was a postdoc, I was a TA at Cold Spring Harbor with Paul Kammermeyer at a patch clamp course. We taught people how to patch clamp, and so I stayed friends with everybody that was still here. And I asked him to invite me to come give a talk. I did, and that's how we had this conversation, and ended up talking in Charles's office. We had slated like 30 minutes, and I had turned my phone off so that I wouldn't get interrupted, and we went like 90 minutes or something like that. And I almost missed dinner. But it was just great. And Charles asked me what was my plan. And, I was like, I'm gonna get a job. That's my plan. And I didn't have any prospects, right? I had applied at that point for almost probably 100 industry jobs.
Okay.
Didn't get any. I had two real interviews there. Kind of had just said, okay, I'm going to give up this academic dream and do industry, and then came here, and things 180'd, and it was really nice. Yeah.
Therein was opportunity for us, and the proof was that when John got here, it just took a little bit of fertilizer, water, and sunshine, and bam, he was right on it right away. I mean, he was starting to fire on many cylinders, in the first month or two that he arrived. That's the energy that he brought to it, and so having success in fundraising and in publications and things like that. So we got this affirmation back right away that it was working out well.
Great.
Yeah, I wasn't going to screw that up. Yeah.
We had Makaya Papasergi-Scott on not too long ago.
She's amazing.
Amazing. She, I think, is newer than yourself.
Yes.
But we, it came up during conversation, just interesting things that come up when you first start a lab, that she didn't think about.
Yeah.
Anything like that you can speak to, of like starting a lab, what that was like, and things that maybe unexpected things you didn't think you'd be learning, or
I gotta be honest with you, and this sounds like people can't believe that this is the case, but a lot of people come in and they have what's called like imposter syndrome.
Yes.
And they're like, oh man, I got this thing, but am I good enough to do it? And I did not have that. I didn't. And it's not because I'm arrogant I just had been through so much that I was like, yeah, I know how to do this. And I think so. And I was ready. I think I was ready for many years. And I think that the thing that I knew I had to do first was your first hire is super important. And I focused on getting a really good technician before I would get a graduate student, because I knew quickly it was going to become overwhelming for me. So I hired Katie Edwards, now Katie Lupia, who was the rock of our lab, and I don't think that she quite expected what the lab was going to be when she signed on for this. The lab was small. There was just the two of us, really, an undergraduate maybe. And we were sharing my desk. And I always say, it was like Billy Joel and Elton John like face to face. And that's what, and we just answered emails, but she's been just tremendous in getting everything set up. So I think that while I think I had vision for this, it was really the people that I hired early on that has made me successful. So Katie Edwards, and then it was Joe Porter, who's a postdoc and now a research assistant professor in the lab. And without them, I would not be successful. So I think it's the people you surround yourself with. And you should always be surrounding yourself with people who are smarter than you and who are good people. And once you start doing that, it kind of becomes easy, you know? Yeah.
As we record this, we're somewhat fresh off of Graduate Student Appreciation Week, but just made me think of, how we kind of pride ourselves here at the institution on being graduate students drive the research mission here. Can you just speak to the importance of them? And
I mean, I talked about Katie Edwards and Joe Porter, but my first two graduate students were Rory Coe and Lily Cisco, and they're tremendous. They work so hard. And, I think my goal as a mentor is to be like a player coach, is not to be, hey, you do this, you do that. It's to be, boots and, with them trying to figure this stuff out and giving them respect. Yeah, they're tremendous. Each of them have worked really hard, figuring out problems, and each of them have been really successful in my eyes. Rory Coe went off and is now in industry, and Lily is actually still with me. She's a postdoc. For personal reasons, she wanted to stick around. And so it's just wonderful having her in the lab and everybody else. I want to make sure that everybody gets covered here. But yeah, they were my first two graduate students. And also, Rochester has amazing undergraduates. And so I brought in really amazing undergraduates over the years, and they bring a lot of energy and keep everyone young. They work really hard, not afraid to get their hands dirty with, kind of the day-to-day stuff in the lab. So they do a lot of the things that are, not really maybe elegant in mind.
But are willing to do those.
Yeah, I mean, the most important thing that I always say that anyone in the lab has to do is genotype animals. It's like it's one of the most important things, and people come in and they do it, and they do an unbelievable job.
And Charles, obviously, you had a great experience with John, but anything else to add there as far as grad students or, and just students in general kind of driving the work here at the institution?
My experience training, much less than John's in terms of graduate students. And I've only been able to manage one by one, not several at a time.
Yeah.
But much in the same way. And the students who found their way into our group were people who wanted that kind of team science enterprise, who had a therapeutic bent. And each one of those people, I think, has had impact on the field larger than they expected when they started. Some of our graduate students, by the time they finished, they were sitting at the table at meetings with people at biotech companies who were designing clinical trials and giving them advice on how to design their trials. So it was a good experience for them.
That's awesome. And John, you are, heavy background in basic science.
Yep.
And now here you are, mastered pharmacology, drug design.
I haven't mastered much.
Diving right into that world for trainees who might have that basic science background but want to make that crossover. How do they cross that line? And interfacing with the world of clinicians.
Yeah.
And medicine.
I mean, I think coming to Rochester really helps with that, to be honest with you, because, you can walk down the hall and I walk down the hall often to talk with Charles, but it's a lot of it's just ease of access, right? And putting yourself out there. Another thing is, which was super helpful for me, when I did my postdoc, it was a great environment for interacting with patients. You would go to meetings. I went to a lot of patient-oriented meetings and interacted with the families there. And I think that for me, that's what's driving my research now. I remember my first Myotonic Dystrophy Foundation meeting I went to. Charles was there. and you talk with families, and they tell you about what's happening with them, their quality of life, different scenarios, how the world works with them in it and how they work in the world. And it's really shaped my desire or my path in research. I really want, I want to help develop therapeutics to help those people. And I never thought, I never thought I would be this far into it. I always imagined myself in a lab where I'd be doing way more basic science. And so the research I do, the things that I'm interested in, I think I'm fortunate because there is a translational slant to it. There are people that do the things I do and don't go that way, but this gives me opportunity. And I don't know if that's the reason I picked them or why I'm interested in them, but certainly it's giving me opportunities to do that.
And you may have answered this already, but that talking about patient interaction made me think of Eric Wagner, another professor here, and a video he recently participated in through our Center for Advanced Research Technologies, where he talked about a story of his, had never met anyone with a BRAT1 mutation, which he focuses on. But his wife, I guess I think in the video he says, we're sitting watching TV, and she's mentions, she's scrolling social media, and she's like, a kid at my school has a BRAT1 mutation. And, then I think one thing led to another. And it just, once you meet or interact with someone that is, experiencing life through this, this condition that you are, trying to help with, it makes quite a bit of difference.
It gives you context and purpose, I think.
Yeah.
Charles, you've told me this with patients that you interact with. This is what you think about. Also, you can learn a lot by just seeing a patient. We study muscle weakness, and you can see their hand, how they're working through picking up glasses, and you're like, well, that's a problem I need to fix. Just to be able to turn a doorknob to get out of, or to drive, or there's things like you don't need to cure a disease. I think cure is tough, but treat symptoms even, I think is really important. And I think about that a lot now. And there's, I've been very fortunate to interact with different patient families. So through the Cystic Fibrosis Foundation and Emily's Entourage and Myotonic Dystrophy Foundation, MDA. So there's lots of them out there. And I think it's the most rewarding part of those meetings.
Yeah, I think in some of our earlier meetings, you mentioned working with organizations like that, as opposed to you're getting funding from organizations like that. You're working for that cause, or you see the cause. How does it change your mindset at all, if at all, getting funding from places like that?
Yeah, so the idea of advancing the field or doing what you say you're going to do. So when I get money, very fortunate to have money from different foundations, is that when I write down we're going to figure this out, I work on that. I don't become unfocused. And so, I sit and I think, how they raise money to do this research is they were out doing walks on the weekend. They were giving up their own money to give to these foundations. They spent time for something that they care about, and I'm going to do the same. So I really make it my best effort to follow through. And so,
I think that is a good reminder to just anyone in the public, that made a connection for me. Yes, you see these 5Ks for XYZ or these walks for XYZ. The money, some of that money is then going toward folks like John, who are dedicated to treating a symptom or.
Yeah, absolutely.
Yeah.
And so I take it very seriously.
Yeah.
And I think you should. Yeah.
You've mentioned that, we're talking about funding. It's important, but also can be some of the more stressful parts of this job.
Yeah.
How do you manage that sort of, I guess, constant stress of got to keeping this work going?
Well, I think Charles has been far more successful than I have. I often ask Charles, like, how have you done this for so long?
What do you think, Charles? How would you answer that?
It's to string together a series of unlikely events over the course of 25 or 30 years. It sounds like it might be impossible. It's not. Okay. And it is possible to keep the momentum going, and you just have to keep building and making progress every step of the way. But there's plenty of people around who are doing that. I think it's a discouraging time right now, and that people who can weather this storm are going to be in a good position to be making big contributions for a long time into the future. So.
Yeah, I agree with that for sure. It is stressful now, and I think, what keeps me going is, one, I have questions I want to answer, but also, I've taken on a responsibility of having trainees in my lab. People who have bet on me to support them, and I take that very seriously. And so, we have lab meeting every week, and I look across the room. It's like, oh, I gotta feed and keep these people. I gotta keep these people employed, and so that that stresses me out. But I also think it gives me fire to write these grants and not become complacent, even when funding opportunities are few and far between.
And you've diversified too, I would say.
Yeah.
Is that, would you say? Not just federal government funding, but other sources, foundations you mentioned, and work with people who are in industry too.
Yep. Yeah, and I think you want to keep all those opportunities, open. I think pursue all of those, and they're not all going to be winners. Some of them will, and you don't need a winner every day. You just need to have them kind of overlap a little bit. So hopefully you can get your funding, 2 years of funding and take care of the gaps, and then like have your major funding fill in everything else, right? And I think it is a never-ending thing, but there's also writing grants is the best way to flesh out ideas. It's not until you put your pen to paper where you realize that was a really stupid idea. There's no way we can actually accomplish this. And actually, Charles and I were talking last week about, let's just talk about our specific aims. What are we trying to accomplish here? Let's try to focus in on this and sort our ideas out. And it's super helpful to talk about the ideas, write them down as soon as possible, and then get going on it. And working with Charles on my first couple of grants was super helpful. I think that every young investigator should have a Charles Thornton. It's someone who's going to say, that's a good idea, or that's a good idea, but maybe let's not include that in the grant, or let's focus in on this. And I think having mentors like with Charles and Bob Dirksen have been super helpful for me for getting funding. I wouldn't have been able to have done it without them, to be honest with you. So it's not obvious writing grants. It's, you think you want to put all your ideas down, you want to wow them, but there's also a bit of how about you write a good grant, and one that is going to get reviewed well. And so that has been super helpful to me.
So follow the guidance, write it in the way that is supposed to be written. And yeah, absolutely. And that goes a long way.
And I've given talks on this. It's now is not the time to add pizzazz and sparkles. You want to it's there's a format, and you follow the format, and you put the best science in there you can. And I think actually Charles was, even how you state every sentence. It's I think about it now, and for better or for worse, getting out the thesaurus, what is a double meaning to this word? Charles really instilled that in me. It's I think that this sentence means this. It's oh, I guess that's true. And you don't know who's reading your grants, and so you want to make it so that anyone who picks it up, they get the meaning that you intend it to be. And so it's painful sometimes, but it's super rewarding in the end. So
yeah,
I think it's been, we've worked well together that way. I think based on your experience, as a clinician trying to move into proposals that involved basic science and genetics, I also encountered headwinds.
Yeah.
And as did you.
Yeah.
And so you kind of learn a posture to deal with that. And it's trying to, basically, in a nutshell, trying to make it as hard as possible for someone to easily step on you and discount what you're doing. Mm-hmm. And so trying to defend against that skepticism at every step was always part of it, I think, for both of us.
Yeah, absolutely. Super helpful.
Yeah, I think that's great practical advice for, trainees
Mm-hmm.
that may be watching this and in the process of writing something like that.
I mean, so Charles just won the mentor award,
Yes,
2 weeks
that's
ago,
right.
last week.
Mm-hmm. Mm-hmm.
This was part of that, right? It was I wrote grants, and then he would pour over them and say, I like this, I don't like that, and you need that. No matter, even now, I've written a lot of grants. Like the last RO1 I wrote, I was having a hard time putting two things together. It wasn't even on something that Charles does. It's on the tRNA work we do. And so it just doesn't sound like you need that in there. And, it was like the last week before submitting this, and I deleted it, like an entire aim. And it ended up being the right idea, right? So it's really good to have a sounding board that you can, pass your ideas by. You don't need to take what they say. You just, you can say, well, no, I'm not going to do that. But at least people are giving you advice that work for them, and it's been super helpful. Yeah.
You mentioned team science earlier, collaboration. You mentioned before that most labs, you don't have all that expertise in just one lab. So collaboration is key in that regard. You mentioned a story of being at a cystic fibrosis conference.
Yeah.
And kind of showed the power of networking. Can you talk a little bit about that story and kind of just emphasize the importance of team science a little bit?
Yeah. So, this was actually because of a funding call. The Cystic Fibrosis Foundation have these collaborative grants. And I was working with Professor David Bedwell down at University of Alabama, Birmingham, and we really get along. And this was on some of our tRNA work and coming up with all these ideas. And we wanted to put in an application. But the somebody at the foundation was like, well, I think you need someone else. This isn't going to cut it. But I've been interacting with this person at University of Michigan. They're really excited to get into cystic fibrosis. How about I link you up with them? And so that was Patrick Thibodeau at the foundation hooked us up with Kristen Kutmo at University of Michigan, and we after our first, it was like speed dating, But after our first conversation, we're like, man, this is amazing, they are an amazing RNA biologist. David Bedwell is he's done just a ton of work in the nonsense mutation space, and I'm a guy. And so but we hit it off, and we put together a really good collaborative grant, and we're going to continue working together. So it's going to meetings, interacting with people, and maybe even meet in conferences where it's not really what you focus on. I think you can get a lot of ideas from other fields and bring them into your own, or collaborate with people doing other science to advance yours. I think there's a lot of science that's siloed. Mm-hmm. And, I see this in muscle research and in lung biology, and, muscle is advanced quite a bit with delivery of therapeutics. And I think you can use these ideas in airway and vice versa. And I think it's just really good to cross-pollinate in that way.
About to wrap up here. This has been an awesome conversation. Really appreciate you guys being part of this. Charles, we'll start with you on this last one. Just kind of any final piece, parting advice for trainees watching this that are either about to hit the job market or navigating graduate school. Anything you want to leave us with?
Probably most of your audience are people who aren't particularly thinking in terms of therapeutic implications of their work or making therapies. I understand that. But for those who are looking for an environment where you are mixed, John mentioned how things are mixed up here. and clinicians and basic scientists get thrown together a lot, and they interact a lot. They're not in different buildings. They're often neighbors in the same hallway. And I think looking for that, if those are your interests, is a good thing. And something unusual about our environment that I see being replicated more in other places now successfully is trying to have a connection all the way through from the basic science discovery work and therapeutic development side right through into the clinical arena is very synergistic and powerful now. And there's a amazing confluence of strong tools on all sides of that equation, and having them together in one site is something to look for if you can find it.
It's something that sounds like sounds like something that's catching on, but it sounds like it's something like that just makes sense. Like it should be that way, right?
But they were so separate for so long.
Yep.
Yeah.
And there's, campuses are set up where, you have the hospital and then you have the research center. And, and I think that just by happen chance, Rochester set it up correctly, back in the day, and it's really worked out. And it's hard to, if you've already gone down that route of separated clinical space and basic science, it's really hard to fix that. And so, but yes, I think we're fortunate for the work that we do, that, we're down the hall from each other and we can actually see, work together in that way and not have to get on a bus to go and interact.
Then you can see how it doesn't happen a lot.
Yeah, exactly.
It is a lot.
We're all so busy. It just takes a little bit of a barrier to stop a conversation from happening, right?
John, take us home here.
Yeah.
Any final parting words?
I think it's a difficult road to get to where I am. But now that I'm here, it's super rewarding. Working with trainees, I have colleagues that I can walk down the hall and talk to. People who want to see you succeed is really nice. I remember the first time I wrote a grant, it was an R01, and I came back all kind of like, oh man. And I showed it to Charles, and he was like, don't worry. I think you said I'm not going to let you die in the vine. And I was like, oh, okay. Well, I got somebody here who's going to make sure I'm okay. And then the next time I brought it by and I had a really good score, he said that put glad in his heart. And so I was like, okay, I have somebody who's wants to see me succeed and will try really hard. And I think, I'm very fortunate. And I hope that people can identify these people to help them advance their career. Just be friends, really.
I love it.
Yeah. So,
all right. Well, I think that's a great way to end it. Charles, John, thank you both so much for being part of it.
Yeah.
Thank you.
Thanks so much for tuning in to the latest episode of the Next Step podcast. If you liked what you heard, please share with other members of the University of Rochester community or anyone you think could benefit from these career stories. And we wish you nothing but the best in your own career journey. Until next time.